Imagine waking up each morning and just like checking your social media updates, you use a smart commode. And this isn't just any toilet. This futuristic commode doesn't just handle your business, but it is a high tech device that analyzes the type and amount of bacteria in the stools you pass. And based on this real time data, it gives you personalized advice on what to eat, what supplements to take, and even suggestions for your daily activities. It's like having a health coach right in your bathroom. This might sound like science fiction, but the technology to study our microbiome in such detail is rapidly advancing. We are talking about genomics, transcriptomics, proteomics, metabolomics, and so on. These cutting edge fields are unlocking the secrets of our microbiome and paving the way for personalized medicine. Welcome to episode 2 of Microbiome Magic. I'm Dr. Jyoti and I'm deeply passionate about this topic. Today, we are diving into the cutting edge technologies reshaping microbiome research. We'll explore how these tools work, the profound insights they offer, and their potential to revolutionize healthcare. In the past, studying bacteria meant waiting cultures to grow, and this was very time consuming and it didn't capture all the bacteria present in the sample. But now, thanks to next generation sequencing techniques, we can quickly analyze the genetic makeup of all the organisms present in a sample. And this is a game changer. Remember the Human Genome Project? It took 13 years and thousands of researchers all over the world, plus $2.7 billion to complete. Today, with modern technologies like the Illumina's new sequencing methods, we can sequence up to 20,000 genomes per year, which means that we can sequence a person's entire genome in less than a day and for less than $1,000. This remarkable progress has made genetic testing easier, cheaper, and more accessible than ever before. After the Human Genome Project, we realized something fascinating. Humans don't have enough genes to account for our complex biology. So that's when the Human Microbiome Project kicked off. The idea was that if humans don't have enough genes, maybe the microbes living in our bodies do. This realization is crucial. A big part of our functionality comes from microbial genes and not from our own ones. So, when we neglect our microbiome through antibiotics, processed foods, poor lifestyle choices, or exposure to toxins, etc., we risk losing these genes and their capabilities. And this compromises our overall health. Think of our microbiome as an ecosystem. This ecosystem has evolved to live in harmony with us. We provide a home for these microbes, and in return, they play a vital role in maintaining our health. But when we bombard this ecosystem with antibiotics, with processed foods, lack of sleep, lack of exercise, and all the poor lifestyle choices, it disrupts this harmony. And it is like turning a very supportive friend into a potential enemy. 70% of our immune system resides in our intestinal lining in the gut associated lymphatic tissue. And this immune system is in constant communication with our microbiome. When there is dysbiosis or a dysfunctional microbiome, the immune system is the first to get affected or dysregulated, and this leads to chronic inflammation. And we now know that this chronic inflammation is the underlying cause for several of the chronic diseases in modern world, from metabolic diseases like diabetes and heart disease to autoimmune conditions, mental health disorders, neurodegenerative disorders like Parkinson's and Alzheimer's, and even cancers. So before we dive into the specifics, let's get clear on some definitions. Microorganisms are tiny organisms that can be bacteria, fungi, archaea, protozoa, or viruses living in all kinds of environments. The term microbiota refers to a community of microorganisms in a specific environment. For example, we have the skin microbiota, the gut microbiota, the oral microbiota, and so on. And then there is the microbiome. The microbiome includes the microbiota, that is a community of organisms living in the whole body, along with their genes and the metabolites they produce. As we discussed in our previous video, the number of genes in our microbiome is 100 to 150 times the number of genes in our own human genome. When microbiome research first started, the focus was on individual strains of bacteria and how they functioned. But now we have come to understand that these microbes operate as a community or a guild. Within this community, there is constant communication between different strains and species. They exchange information about our internal and external environments. Imagine they are all part of a massive chat group, constantly messaging each other about what's happening in our bodies and what actions to be taken next. So, this intricate network is our microbiome. Ultimately, our traits, behaviors, and health conditions result from the interaction of this microbial community with our genetic makeup or the genotype and our exposome or the environmental exposures. This dynamic interaction shapes our phenotype, which is the observable characteristics that define who we are and how we feel. So, what exactly are genotype and phenotype? The genotype is like the blueprint of your body, the set of genes you inherit from your parents. Think of it as a recipe book or a cookbook. On the other hand, your phenotype is your unique set of physical traits, behaviors, and even some of your health conditions. Basically, your phenotype is like a finished dish or a curry, and your genotype is the recipe for the curry. But here's where it gets really interesting. The world around you, everything from the food you eat, the air you breathe, how well you sleep, the stress you are exposed to, and the toxins you encounter, all these can change how your genes work. And your microbiome plays a big part in this process. So your microbiome chats with your cells and can influence which genes are turned on or off. For example, even if you have inherited genes for breast cancer from your mother, you might not develop cancer until much later in life or maybe not at all. The timing or whether you develop it at all depends on your lifestyle, habits, and environment, which in turn depends on the interaction between your environment and your microbiome. So while your genes set the stage, the environment, including your microbiome, helps to write the script of your health journey. So now let's see how our genotype, the genetic blueprint in the DNA, transforms into a phenotype, which are the traits and behaviors that define us. The way information in our genes is turned into these traits is similar in all living organisms, including microbes and humans. And this process is the foundation of genetics. Imagine each microbe in our microbiome as a tiny chef armed with its own cookbook, its DNA. This DNA acts as a comprehensive set of instructions detailing everything the microbe needs for survival and function. Just like these microbes, human beings and other organisms also have their own cookbooks in the form of their DNA or their genotype. And this guides the development of all the traits and behaviors of these organisms. This process is fundamental to understanding genetics in all forms of life. So, the first step in the journey from the genotype to the phenotype is transcription. Picture the microbe's DNA cookbook filled with countless recipes. When the microbe needs to make a specific dish or a specific protein, it doesn't take the entire cookbook with it. Instead, it carefully copies the specific recipe it needs onto a notepad. This notepad, known as the RNA, now holds the precise instructions for what the microbe intends to produce. The next step in the journey is translation. The microbe takes that RNA notepad filled with the instructions and sends it to its kitchen, the ribosome. Inside the ribosome, the RNA is read like a recipe guiding the assembly of specific proteins. These proteins are the functional units that drive everything from enzyme activity to tissue structure in our bodies. They are like the finished dishes prepared by the microbe's molecular kitchen, that is the ribosome. So each protein serves specific purposes such as breaking down nutrients, building tissues, or regulating processes essential for our health and well-being. The gut microbiome is like a bustling biochemical factory, not just producing proteins but also a diverse range of metabolites. These metabolites can be hormones, neurotransmitters, vitamins, nutrients, and even waste products. These substances produced by our microbiome support digestion by breaking down nutrients. They regulate our immune responses and even influence our mood and metabolism through the production of neurotransmitters and hormones. By shaping these essential processes, the microbiome plays a crucial role in maintaining our overall health and physiological balance. So, understanding and nurturing this relationship can lead to significant improvements in our well being. This groundbreaking study, published in the journal Science in 2013, dives into how our gut microbiome influences body weight. Researchers took fecal microbiota from pairs of female human twins, where one twin was obese and the other was lean. They transplanted these microbiota into germ free mice. What they found was fascinating. Mice that received microbiota from the obese twins gained significantly more weight and body fat compared to those that received microbiota from the lean twins. In essence, the microbiota from the obese individuals appeared to promote weight gain more effectively than that from lean individuals. This study vividly illustrates the powerful role our microbiome plays in shaping not just our health but also our body composition. So, now let's look at the technologies which help us understand the microbiome better. First, we have genomics. This is the study of the entire DNA blueprint of microbes. So, because of these cutting edge technologies we have now, we can swiftly read and analyze the genetic material from thousands of microbes all at once. It's like having a super fast scanner for our microbial cookbook, revealing all the recipes at our fingertips. Next, there is transcriptomics. This focuses on the RNA, the copies of those recipes. By sequencing RNA, we can see which genes are active and what recipes the microbes are preparing to cook right now. Moving on to proteomics, the study of all the proteins, which are the finished dishes. This stage tells us whether the microbes have actually cooked using those recipes. Advanced technology now allows us to identify and measure these proteins, showing us precisely what the microbes are doing and how they interact with their environment. And lastly, metabolomics dives into the ingredients and the byproducts, which are the metabolites. Using sophisticated techniques, we can now map out the chemical fingerprints of microbial activity. This gives us a detailed picture of what's happening in their microscopic kitchen, from the ingredients they are using to the byproducts they are producing. When we study the microbiome, there are three main questions we need to ask to really understand what's going on. Let us break these down. The first question, who is there? This means identifying all the different types of microbes present in the environment or the sample we have taken. It is like taking roll call in a classroom to see who is present. This is where genomics comes in. By sequencing the DNA, we can map out the entire community and see which microbes are present in that specific sample taken in a particular site. The second question, what are these microbes capable of? In other words, this involves looking at their genetic potential. What genes do they have and what proteins and metabolites are they capable of producing? It is like checking what tools each student in the classroom has in their bag. This step involves both genomics and transcriptomics to see not only the potential encoded in their DNA, but also which genes are being transcribed actively into RNA. The third and the final question, what are they doing? So, finally, we need to know what these microbes are actually doing. To truly understand what our gut microbes are up to, we need to dive into their current activities. Are they producing proteins? What metabolites are they generating? This is where proteomics and metabolomics come into play. By studying the proteins and the metabolites, we get a real-time snapshot of what these microbes are doing. Now, multi-omics integration is a game changer in precision medicine. It combines data from different layers of the microbiome like genes, RNA, proteins, and metabolites to guide personalized treatment decisions based on each person's unique microbiome and molecular makeup. When we integrate metatranscriptomics with metaproteomics and metabolomics, it's like watching a day in the life of the microbiome. We can see how microbial genes, proteins, and metabolites change throughout the day following circadian rhythm and responding to what we eat and do. Now let's talk about some cutting edge tools that are pushing the boundaries of microbiome research. First, we have got spatially resolved sequencing. This fancy technique, like laser capture microdissection, lets us zoom in and analyze microbial communities at specific spots in the gut. It's like having a microscope that shows us who's hanging out, where, and what they are up to. This helps us understand how the microbes organize themselves and interact over time, especially when we change what we eat. Then there is advanced imaging and gut on a chip models. These tools let us watch in real time how the gut microbiota, our bodies, and the environment all interact. Imagine being able to see inside living animals with tools like two photon microscopy. It's like having a window into the tiny world of microbes, seeing how they move and respond to different conditions. Then there are the microfluidic gut on a chip models. These mimic our intestines and let us grow gut microbes in conditions that mimic our daily eating patterns. It's like having a mini gut in the lab where we can study how the microbes interact with our bodies in real time. Stable isotope probing is another method. It's like tagging microbes to see what they are eating and how they process nutrients throughout the day. This helps us understand how their activities change with our daily rhythms and food intake. All these tools together are giving us an unprecedented look into how the microbiome works in real time. We are uncovering the intricate patterns and dynamics of our gut bugs, showing how they respond to things like our daily routines and diets. And these advances are really pushing the boundaries of what we know about the microbiome and how it influences our health day by day. With all this data, the next challenge is making sense of it. This is where bioinformatics comes into play. Bioinformatics uses computer algorithms and statistical methods to analyze and interpret complex biological data. Through bioinformatics, we can identify patterns, make predictions, and generate hypotheses about how the microbiome affects our health. It's like having a supercomputer that helps us decode the language of life. One of the most exciting applications of microbiome research is its potential to personalize healthcare. Instead of a one size fits all approach, personalized medicine tailors treatments and lifestyle recommendations to the particular individual. By understanding a person's unique microbiome, we can pinpoint specific imbalances and tackle them with precise interventions. For instance, if someone lacks certain beneficial bacteria, we might suggest tailored probiotics or dietary adjustments to restore the balance. Companies like Viome offer at home test kits that analyze your microbiome from a stool sample. These tests provide detailed insights into the composition of your gut microbiome, highlighting potential imbalances and offering personalized recommendations to improve gut health, skin health, and even mental well being. This approach represents the forefront of personalized medicine, promising a future where health interventions are finely tuned to each individual's microbiome profile. Similarly, there are kits available for analyzing the vaginal microbiome, which may be useful in those patients with recurrent vaginal infections that are not responding to our usual management. Looking forward, imagine a future where monitoring our microbiome is as routine as checking our vital signs. Wearable devices or smart toilets could provide real time feedback on our microbiome status, offering personalized advice to enhance health and prevent diseases proactively. This transformative approach has the potential to revolutionize healthcare by enabling early disease prevention and supporting lifelong well being. If you enjoyed this video, don't forget to like, subscribe, and hit the notification bell to stay tuned with our latest content. Let's continue to explore, learn, and stay curious together. Thanks for watching and see you next time.